Assalamualaikum and a very good morning to everyone. My name is Azwa binti Azizan and I'm here today to present my final year project titled Phytochemical Analysis and Antimicrobial Activity of Pereskia bleo leaf extracts against oral pathogen. The use of medicinal plant has been well documented since ancient time. This past decade has witnessed a surge in public interest toward herbal remedies. Moreover, WHO encouraged secondary plant metabolite as alternative drug. Originated from the family of Cactaceae, Pereskia bleo is chosen in this study. Despite the number of reviews on Pereskia species, the data on P. bleo, phytochemistries, and antimicrobial activity is still limited and scarce. In order to avoid the emergence of antibiotic-resistant genes and also the use of synthetic antimicrobial chemical, which may cause various health outcomes, antimicrobial agents derived from natural products are needed. This finding should provide a valuable information on P. bleo extract as potential alternative drugs and provide the data on its effectiveness. The objective of this study are to extract the bioactive compound of P. bleo leaf using methanol, chloroform, and anhydrous solvent, to analyze the phytochemical constituents of P. bleo extracts and also to evaluate the antimicrobial activity of P. bleo leaf extracts against oral pathogen. Moving on to the literature review, the emergence of antibiotic resistance. WHO has declared that antimicrobial resistance is one of the top 10 global health crises, and the infection produced by multi-drug resistant bacteria are becoming more widespread and poses a severe public health risk. Next, according to Rehab and Envira in 2019, phytochemicals are described as antimicrobials, protecting plants from the pathogen. Some of the compounds that are responsible for the antimicrobial activity are as follows. Moving forward to the plant, P. bleo is originated from the family of Cactaceae and well known as jarum tujuh bilah in Malaysia. It is a prominent medicinal shrub to treat various ailments such as gum problem, ulcer, and cancer. There are about 11 phytochemicals that has been associated with this plant, and they have significance in antimicrobial activities, which possess antibacterial and also antifungal properties. These are the overall methodology used in the study according to the specific objective. Comply to the first objective, these are the extraction process used. For the second objective, the phytochemistry analysis were done using various methods as follows. Lastly, the antimicrobial activity was screened using the diffusion method, MIC and MBC assay, Bioautography assays, and synergistic impact of crude extract using this diffusion method. For the result and discussion, firstly, the yield of extraction. This study revealed that the yields of P. bleo extract increase with increasing polarity of solvent use. The type of solvent chosen, its polarity, and the extraction technique impact the yields of P. bleo. Next, the finding of secondary metabolite analysis showed that different phytochemical compounds were found in each extract. This could be due to the variation in polarity levels and species natures in the samples, environmental impact, extraction process, and also the analytical method used in the analysis. This diffusion exhibited various antimicrobial activity with the highest inhibition zones of 8.93 mm of chloroform extracts against S mutants. In MIC and MBC assay, chloroform show promising antimicrobial activity against S mutant and C. albicans. The variation in antimicrobial activity may due to the distributions of active compound in the extract. Different extracting solvent had different impact on the chemical distributions and possess different antimicrobial activities. Chloroform was selected for further assay as it produced the best antimicrobial activities compared to other extracts. Next, bioautography of TLC failed to produce any zone of inhibition, hence no antimicrobial activity shown here. The reasoning is the concentration-wise of the separated spot, the extract spotted on the TLC is too little and get diluted as it travels through the TLC. This finding deduces that the antimicrobial activity is due to synergistic effect of compound in the crude extract and not the individual compound. Lastly, the synergistic impact of the crude extract was studied with combinations of 10 micrograms streptomycin. Enhancements of inhibition zone at 34.07% can be observed. Therefore, it is hypothesized that Biblio leaf chloroform extract could be relevant in combination therapy. As conclusion, methanol, a polar solvent, produces a larger yield compared to an hexane, a non-polar solvent. These leaves of Biblio are rich in phytoconstituents as shown in the qualitative analysis. All of the extracts were found to exhibit antimicrobial activities against the tested strain. The effectiveness of antimicrobial activity can be enhanced using a drug combination therapy. Further study, including identifications and purification of the active compound, synergistic impact, and also toxicity studies of Biblio will need to be pursued. Thank you.